Okay, thank you very much for the kind introduction again. And I take it as an endorsement that none of you have run off, that you didn't disagree with me when I talked to you about my idea of confirmations, which is good to see. So here's some more, if you like the first bit. So see if I can bring this up. Well, there was that bit as well. Okay, so slideshow. I would just like to start off with a little disclaimer. I'm going to talk to you about work that we've done as part of a small company. I'm not here as a business development representative. I'm here as a scientist. So hopefully you will see the talk in the light of the science, and not as a particular sales pitch, which is not what it is intended to be. So the outline of the talk is falling basically into three categories. The first one is just a general introduction into the concepts that we're trying to pursue with the work that we're doing, which is mainly around NMR, and the use of NMR data in new ways in order to characterize conformational behavior of molecules. And then I would like to go into the actual application of this particular methodology, and the conclusions will hopefully draw to a close the discussion on conformations that I'm leading today. So, conformational design is a point I already raised in the previous talk, and I would like to put it into a different context now. When it comes to it, what the medicinal chemist is always chasing is the opportunity of influencing a bond to be exactly the match in terms of its torsional profile to the trajectory it needs to be in order to give you good shape matches with the binding site. Unfortunately, as it often is the case, this desired state is not necessarily reflected in the natural behavior of the molecule prior to the binding event. What you see is that torsions are often either multiplied into different positions, or you have a slight deviation between the torsion that is naturally occupied and the torsion that you have in the bound state. So there is an opportunity here in order to change the chemistry to make those torsional changes fit for purpose in terms of the screening, the binding site. And the tools which are at the chemist's disposal are already introduced earlier, but they basically fall into two categories. There are electronics and there are sterics, which we can play with, and there are a multitude of those. So I would like to urge you all to consider these various different opportunities in terms of design of molecules more fully, and consider the impact of those on your conformations of molecules. In terms of what we do in the context of our work is very much focused on the traditional make, test, design cycle that is being pursued in the pharmaceutical industry. However, we have adapted it to the context of conformational information. So we first start with conformational analysis of our molecules, and that is very much an opportunity to understand not just the shape of molecules or the wobble, the torsional flex around these particular torsional states, or indeed the opportunity of assessing their probability of existence, but it's also about understanding the controlling factors that actually lead to all of those that I just mentioned there beforehand. That gives us an understanding of what the molecule is doing, but in order to derive a hypothesis to drive design ideas, what we need are reference points, i.e. what kind of conformational states we are actually aiming at. And they could be either coming from an understanding of the binding mode, a pharmacophore, as we've heard in the preceding talk, or indeed reference molecules that we can use in order to assess how molecules with different chemistries sample chemical space. If they hit the same target, they ought to have a similar shape match if they bind in the same way. Obviously, there are some leaps of faith in that particular statement, but hopefully you stay with me for a while. When you understand what you're aiming for and where you're starting from, then you have an opportunity to derive ideas and points of chemical intervention that are actually going to drive towards the before and after in more closer relationship. And you can use that by, for example, introducing conformational restraints, introducing new conformations in molecules, etc. All of these, however, are going to influence, by and large, the thermodynamic properties of the molecule. But what I'm not proposing here is that when we do conformational restraining of molecules, we are actually eliminating particular energetic terms from the intermolecular pool, energetic pool that molecules have. What we might actually are doing is we're redistributing the internal energy of molecules, because overall the molecules are sitting in a bath of 37 degrees C, which is a significant energy sink. So when we go about doing our conformational analysis, this is actually what we're doing. We determine the conformational behavior of molecules in terms of their dynamics. But when I say dynamics, what we're actually trying to do is build a relationship between the before, i.e. in solution, and the after, when it is bound to the target. And we have seen good predictive properties of our NMR conformational ensembles with respect to assessing what we call the bioactive conformation. And the bioactive conformation is the conformation molecule has to assume when it is bound to the target. So as you would expect from a thermodynamic perspective and from a kinetics perspective, molecules do have the shape in solution that they need to have in order to bind to the target in either more or less close matching detail. What we use for this is small molecule NMR, and with this we use the full ensemble of experimental methods which have been developed over the years in terms of 2D and 1D NMR data, whether it's NOEs or even RDCs, in order to characterize what the molecule is actually showing us in terms of information. This is then extracted using software which we have developed. They are basically around mathematical models which are aimed at harmonizing the information which is contained within the NMR spectra of various types to come up with a conformational model that is required in order to satisfy all the experimental data. So when you record an NMR spectrum of a small molecule, you sample all of the conformational states of that molecule all at once, and therefore you have a mean NMR conformational signal. And that's what we're trying to basically develop as a result of untangling all of that information. So when it comes to it, we do not use any bootstrapping of computational chemistry methods in order to preload or preconceive, if you like, the conformational space, and therefore hopefully we do not have any bias introduced in the process. The information we get is summarized here, so we understand the number of torsional positions a particular flexible bond can assume. We can define this in terms of the angle with precision, as well as the movement around that particular position. But importantly, we can also distribute the probability of existence between the various different conformational states a particular bond can go through. So we understand each bond or each flexible element of a molecule, because we include ring puckers in this and hybridization changes, for example, in quite a precise bit of detail. And when it comes to it, the relevance of this can be shown on this slide, because we've been studying now quite a number of molecules for which we have had the opportunity of identifying the bioactive conformer, i.e. the binding mode of the molecule from the PDB. And after correcting for artifacts, we can now understand the relationship between the solution behavior and the bound state. And we can see in all of those instances where we have potent molecules that in solution you see the conformation or a very close analog of that conformational state that is actually required for binding. A second level of validation of the usefulness of this particular way of working is looking at the relationship between small molecule energetic states in the crystal structure, where there are multiple particular polymorphs recorded, and you have seen histidine before, but histidine is a really interesting example, and like other amino acids, there are a large number of polymorphs recorded in the CSD, and they give you an interesting picture of the conformational behavior that histidine might have in solution. But this is what it looks like in solution based on the NMR data. You can see that the Chi1 and Chi2 bonds assume multiple conformer states, and in particular the Chi2 bond is very dependent upon the protonation state, either on the histidine or the amine in particular, and we can talk about that later if you're interested. With the particular orientation come the libration, which is the angle flex, and you can see that all of these bonds are not necessarily extremely tightly controlled, so they all flex quite widely around the mean angle. If you now compare what we have described in solution with what is in the crystal database, you can see that there is a close matching relationship between the solution state and the solid state for histidine. Okay? And this is one piece of evidence we use to argue our case that actually the NMR data and solution data can give you a rich source of information which you can use for conformational analysis that is relevant for biology but also relevant for the solid state of small molecules. So let's assume we have ticked the question of understanding conformations, and even though I've not spoken about it, we do also understand the elements which control those conformational states, so that we can extract that information from physical chemistry experiments, for example, as I've just alluded to with histidine. If we do this twice or 3 times in different chemistries, we can then give our reference points, or we can use X-ray crystallography, and off we go. We can then play the game of restricting conformations. And now into the examples. First I would like to go into the example where we have X-ray crystallography information available, and unfortunately the build disappeared, but when you play the game of structure-based drug design, you have a molecule in free solution, you have a structure of either the protein or the bound state, and you have the before and after, in this particular instant, verified through experimental data. So you should be able to understand exactly how and where to constrain a molecule in order to make it bind appropriately. The other thing you have is you understand quite in detail what the molecule does in solution, and therefore you can use that information in other ways. And this is what we set out to try and demonstrate with this particular project, looking at the interaction of the molecule shown at the bottom, SRS5, against KEAP1, where it binds to, and by doing so it prevents the protein-protein interaction of NRF2 binding to KEAP. We particularly were interested in understanding the relationship of the molecule bound to KEAP and how we can use that information relative to the solution information to understand both the level of interaction between the protein and the molecule, what kind of factors go into it that give you the readout in terms of affinity, but also whether you can use this sort of information prospectively in order to underpin docking studies. And that was not with respect to verifying that you can dock molecules to protein. That was very much with the view of understanding whether the NMR information can actually be used as a means of preempting, if you like, crystal structure determination of molecules of real interest. If you can understand and predict using computational chemistry and the solution structure information, you might not have to go and solve many costly X-ray structures. So in terms of the solution behavior of SRS5, it's a molecule which is extremely rigid. It only has 2 conformer states which are relevant to this side chain of the molecule, and it is partitioned between 85% and 15% of those particular conformer states. I already mentioned it is very rigid, which means that the actual extent of libration, i.e. the angle wobble around the mean position, is really tightly controlled in this molecule. And that is quite special in many ways. This is the most rigid molecule we have studied so far in terms of the actual libration. We have then taken this molecule, for which at the time we didn't have any information of the binding mode because it hadn't been published by that time, and looked at docking studies using just the conformations which we have experimentally verified in the solution to see what we can glean if you use that as input into docking. So what you can see here is the binding site of the molecule that has been defined before from peptide from peptide inhibitors of KEAP1. And we've used that as a start point, even though, and I would like to acknowledge that now we have actually the binding site topology as it was available more recently from work at UCB, where this molecule has also featured as a star in a drug discovery project. When you do the rigid body docking of the conformer states of the molecule in solution in the binding site, you can see that the 85% highly populated conformer states fits reasonably well. And it actually leads you to predict that that is the binding site, the binding mode of the molecule in the bound state. So if you're just taking the mean positioning of all the bonds, you can see that this is what the molecule looks like, and you can use that as a start point for further investigations. As it now happens, we have the actual bound state, and this is the crystal structure of the molecule and its descriptors in terms of the hydrogen bonding interaction. And there's a very interesting bridging water molecule there. Whether or not it's important for binding, It's a matter for debate. I don't think it is, but it's certainly something which is there, and I don't want to hide it. When you now compare the solution structure, which is in green, to the X-ray crystal structure of the molecule bound to the protein in gray, then you can see that there are very many similarities, but there are also some key differentiation. And this is something we are trying to understand at this point in time a little bit more closely, because if you're looking at the molecule in its bound state, you can see very easily that there is a significant deviation, and that deviation is beyond what you actually see in terms of motion of the molecule, and as such, what it actually takes away is the opportunity for the molecule to assume a relaxed position within the binding mode. And we use that as an initial hypothesis to argue that the reason why this molecule, which is a really good fit from a topology perspective and from a hydrogen bonding perspective with the binding site, is only 1 micromolar in terms of its affinity. So perhaps the strain, the induced fit that is required to satisfy all the requirements for binding is what actually reduces the overall binding affinity of the molecule. So when it comes to it, I think we have started to understand a little bit how to use solution structure in the context of in silico prediction of binding modes, and we have been able to understand the relationship between bound and solution, and therefore we have been able to square the loop that I originally introduced in the beginning. And we started to explore this in much better terms now in ways which so far we haven't been able to do, because we are able to see the before and the after and build a relationship which is obviously informing chemistry a lot. So the second example, the one I would like to briefly discuss, is GPR 119, a project we ran right at the beginning, and when we looked into the use of this technology. The receptor binds a lipophilic acid, and there are compounds in the literature which are described to be competitive inhibitors, competitive to this particular agonist, and they also show significant agonist activity. There are also some pharmacophoric models described, but they are based on looking at conformational assessments of the small molecule. As this is a GPCR, there is no crystal structure published or known to my mind for GPR 119, so this is an entirely in silico approach to charting out a pharmacophore. And there are since then other ones published, and they either agree or disagree with that one. Our work was started by the use of a number of the molecules which were described in the literature as good agonists at this particular target. And here are just 2 examples. In total, we looked at 6 different chemotypes, but they all cover the same area of chemical space, and we believe that they're all competitive with each other. And the reason why we believe that is that they have common features within the framework of the molecule. So they have hydrogen bond acceptor groups, and they have lipophiles, but they also have differentiating chemical entities within the framework. And we were trying to understand what the reason is, that even though you have a lot of common features, you also have some different features, but the differentiation is not reflected in the biology. So what is going on? When you look into the actual conformational behavior of these particular molecules in solution, you can see that the BMS compound is reasonably rigid. It only exists in 2 conformer states, and this is simply by chance that there are again a split of 85-15. So this has got nothing to do with the fact that this is the same numbering as for the NRF2. That's just coincidental, and I should have picked a different molecule. Anyway, so there are two forms, either the extended or, if you like, the bent form of this particular molecule. And overall, the motion described in solution is quite extensive, but nonetheless, it groups around 2 particular shape matching areas. I said before, we understand the conformational features that lock molecules into particular conformational behavior, and we believe that for the BMS compound, it's actually the core of the molecule that is important. And we believe that each of these heteroatoms, the oxygen and the 2 nitrogens, they are very pivotal to exactly setting the molecule up to have the right shape for binding. We have not been able to talk to BMS about it, but we would be interested to understand if you're taking the nitrogens out of the central core, whether you start losing agonist activity or efficacy of the molecule. But that's what we believe is happening. So you need each of the heteroatoms in place, and you need the bridge in order to rigidify the molecule to the point where you have the right shape. But what is the right shape? The right shape becomes evident when you start looking into a second molecule from the series, and this is one I picked because arguably it's a lot more flexible in the core of the molecule, but when it comes to it, the actual shape diversity is really quite restrictive, so it is actually really quite linear in its topology. And when you start to overlay the BMS and the Merck compounds onto a single reference frame, this is here the piperidine with the lipophile, you can start to see that there is actually part of the overall shape which is morphing quite nicely together. And if you do not just reference framing on one part of the molecule, but look at the similarity in terms of chemistry, you get this overlay whereby you see that the hydrogen bond acceptor groups, which I highlighted earlier, really closely track each other independent of the template. What is different, though, is the connectivity, the chemistry in between, and the way that the molecules wriggle about, going from one end to the other. But we've seen the diversity of the chemistry that is actually allowed, and overall the consensus shape very much confirms a hypothesis for a pharmacophore model, which has now subsequently been confirmed with more recent pharmacophore model work that has been done. And finally, the third example is one which we've done in collaboration with AstraZeneca, where we studied a significant peptide that they were interested in as an agonist for a GPCR target, and this is GPR103. The work is published, so I can talk about it. And the key point here is that the peptide itself assumes in the key region for biology only a small number of conformer states. And what colleagues at AstraZeneca have done is they have used molecules from the literature to define pharmacophore points through in silico screening methods in order to understand which of those 8 shapes that the peptide is using are actually the important ones for biological activity, and then use that again to refine their chemistry in the project. So overall, the key pharmacophore points were mapped against each other at the C-terminal end of the peptide, because that's the biologically relevant one, and it was also cross-matched against some literature compounds which were used for the same study. When you're looking at the results, the conformational search identified conformers of the query molecule D, which were closely matched to the solution conformer state of the peptide. So naturally, the focus of attention then shifted towards that particular area of chemical space, which there was the overlap for. And what you can see when you do rigid body overlap between the solution conformer states and the most relevant conformation from in silico searching, you can see a really close match between the tracing of the pharmacophore groups across the C-terminal end of the peptide, and thus validating the approach using the peptide structure to tease out what the conformation is that you need to chase in chemical terms, and also at the same time the opportunity for additional affinity building groups, whereby you can see which parts are not mapped but are important for activity potentially. Overall, what was interesting to see is that the conformer, which is a close match to the pharmacophore in solution, is one which is reasonably close in energetic terms and therefore should be accessible, and therefore the whole thing seems to be making quite good sense. Based on some of the work that was done, molecules were then derived, and at the end there was a really interesting piece of work emerging from this, which led to a compound which actually went close to the clinic. So when it comes to it, I now want to conclude by saying that, yes, flexible molecules are partitioning into particular reference conformers which are going to be used and useful in order to furnish ideas for hypothesis formation and therefore medicinal chemistry design. And that is hopefully something which you have all seen now. So in the end, I would just like to acknowledge the people at C4X, the colleagues at AstraZeneca, and some of our collaborators which we have been working with in order to get the compounds made and tested. Okay, thank you very much for your attention.